Hello everyone, welcome back. This week we'll be covering the human nervous system part 2. You can find the information from this lecture in chapter 2, Julian's Drug Primer, pages 48 through 67. Now remember that I don't cover everything that's in the textbook. The textbook is very heavy in biology, and since this is an introduction to bio type of class, I figured we didn't need to know every single detail that is covered in the book. The key concepts that we will cover today are describe the major function of the two types of brain cells and recognize a neuron and a diagram, explain what an action potential is, summarize the activity that goes on at the synaptic cleft, identify and describe chemical neurotransmitters of the brain. And so there are two types of brain cells, and we're going to talk about that first. Just to summarize the key concepts once again, we're covering the types of brain cells, action potentials, synaptic cleft and synapse, and neurotransmitters. So let's start off with the two. There are two major types of cells in the nervous system. One group is called glia, and the other group are called neurons. Glia are the cells, the auxiliary cells that pretty much hang outside of the neurons, aiding in the propagation of signals or providing nutrients and cleaning up dead cells around the area. And the neuron are the longer cells that have a cell body. They have dendrites as well, and this is where they receive signals from other neurons. Signals are sent down the axon to the axon terminals, and then a terminal forms a synapse with the dendrites of another neuron, and this is how neurons pretty much propagate signals to each other. And so again, glial cells are basically a class of cells, and so there are different types. Schwann cells you mostly find in the peripheral nervous system, and these form like these sheaths, kind of look like hot dog buns around the axon, and this helps propagate the signal so that the signal doesn't leak. And oligodendrocytes, those are mostly found in the central nervous system, provide the same type of functioning where it prevents the signal from leaking when neurons are basically sending electrical signals to each other. And so you can think about these type of neurons, and by the way, these what they're doing is forming a sheath, so they're forming a myelin sheath around the neuron. But to better understand that, you can think about when you plug in your cell phone to a socket, and so there's an electrical cord, right? But the electrical cord is insulated with some type of plastic on the outside, and that plastic keeps the signal where it's supposed to be, propagating along the wire, and it prevents you from getting electrically shocked. And so that's what glial cells do. Glial cells provide what you call an insulation so that the signal doesn't leak. All right, so other types of glial cells are astrocytes and microglial cells and ependymal cells. And so all of these also help in supplying nutrients and oxygen to the main neurons and destroy and remove dead neurons as well, and help hold neurons in place. Let's cover the types of glial cells once again. Oligodendrocytes and Schwann cells are the ones that form the insulation layer around the axons. Oligodendrocytes are the ones that are found in the central nervous system, meaning in the brain, whereas Schwann cells are mostly found in the peripheral nervous system, so that's the rest of your body. Like, for example, the neurons that are attached to your muscles to help them move, and also parts of it are in the central nervous system as well. And those are the cells that form like these hot dog buns around the axons to help prevent the signal from leaking, so they provide insulation. Astrocytes play a role in providing nutrients and structural support for neurons, as well as helping in recovering from injuries. And microglial cells are like the immune system cells of the brain, which clean up damaged cells and debris and prune neurons. So neurons aren't really being fired that much, basically are pruned. And research has shown that too much pruning, so getting rid of neurons like at a high rate, plays a role in Alzheimer's. And we know that in Alzheimer's patients have memory loss, and so it can be tied to memory loss. Neurons are the other major type of cells of the brain. There's actually more glial cells than neurons, but yet it's another major type because the neurons are the ones that actually propagate signals to each other. And this is how communication goes on from the brain to the body and from the body to the brain. And so we consider them the electrical wiring of the brain. A neuron has a cell body, just like a regular cell. It has a nucleus where it has DNA and mitochondria to create ATP for energy. It also has ribosomes where proteins are made, and Golgi apparatus where proteins are packaged. And so all of that is very important for a neuron because neurons are responsible for making neurotransmitters, and those neurotransmitters are the ones that emit signals from neurons to neurons. So the anatomy of a neuron also has dendrites, and so the dendrites are these like long branchy areas here, and these dendrites receive signals from other neurons. They actually receive signals from the axon terminals of another neuron. So a signal is received at the dendrite. It goes down the cell body and down the axon, and so the axon is this long part of the neuron, and then the signal goes out the axon terminals. Now remember these cells here. So this one right here showing you a Schwann cell. These are the glial cells forming like that hot dog bun looking thing around the axon to prevent the axon from leaking the electrical signal that is being sent down. Now neurons form tracks. These are like roads to different areas of the brain, and that's how communication happens from different hemispheres of the brain. And then that communication eventually elicits some type of behavior, whether it's a physical behavior or an emotional behavior or thinking. Every single function that you can do is because of all of the neuronal communication going on in the brain. And so, as you can see, there are many different types of tracks. So, you can consider them like roadways. And so, all of these different colored neurons here are just examples of different types of tracks, meaning that this is how neurons communicate to different areas of the brain. Now let's talk about action potentials, which is basically the signal that I was talking about earlier, how I mentioned that the signal is received from the dendrites into the cell body, down the axon, out the axon terminals. Well, that is an action potential, and that is how neurons communicate to each other. And so action potentials are the electrical signals produced by neurons. Electrical signals allow neurons to communicate to each other and to communicate from the body to the brain and the brain to the body. And a neuron, once again, sends information down the axon away from the cell body, so it never goes like from the axon terminal up the axon to the cell body. That's not the way that the signal is propagated. It's actually going down. And so here, if you want to take a closer look, information is received from the dendrites. And where is this information coming from? Well, let's pretend like there's another neuron right here, and that other neuron would actually be pointing its axon terminal to it. So you would have the signal going down the dendrite to the cell body, down the axon, and it's kind of like signal's kind of like hopping. Really, it's not like just a straight line. It's an interesting way in which a signal is propagated. But I guess it would be a little bit hard to explain without showing appropriate pictures from that. But it's called saltatory signal. Anyway, it goes down the axon, out the axon terminals. And we'll talk about exactly what goes on here in the axon terminals very shortly, which is called the synapse. Now, why are action potentials generated in the first place? Like, how is it possible that neurons can generate electricity? Well, it has to do with the types of charges that exist inside of the axon and outside of the axon. And so I don't need you to memorize necessarily what types of atoms or elements are going in and out of the neurons, although I can tell you that it's sodium and potassium that cause these major changes in charges inside and outside of the cell. But it's the actual separation of the charged ions that it's a separation of charged ions that provide the energy to create a voltage, basically. And so, as sodium ions rush out and potassium ions rush in, it causes a how do you say, like a switch in charges. And so here on the inside of the axon, it's more negative, whereas on the outside, it's more positive. And it actually creates a voltage. And so it's about 70 millivolts. All right, now let's talk about the synaptic cleft and the synapse. And so this is the activity that's going on at the end of the axon terminal. All right, so let's talk about the synaptic cleft, and this is where all of the magic happens. This is basically the site of every major drug action. Well, one of the major sites. And I'm talking about pharmaceuticals, like the meds that you take, the meds that doctors give you, and also the site of drug action. So that includes illegal drugs as well. And so the axon terminal is basically the very end of the axon. Remember, I was talking about how the signal goes down the axon, out the axon terminal. And so basically, what's going on is activity, the release of neurotransmitters. And neurotransmitters are the chemical messengers of the brain. Every type of neurotransmitter has a different type of function. And I'm sure you've heard of like dopamine, which is a neurotransmitter made by the brain. Endocannabinoids, those are neurotransmitters made by the brain. Acetylcholine, GABA, glutamate, and we're going to talk about all of those neurotransmitters later. And so once they are released and they bind to a receptor, and remember we said that receptors are found in cell membranes, and receptors can allow the entry of molecules into and out of the cell, or they can just act like literally a receptor, as in they can bind to some type of signal and then communicate that signal to the cell, and that cell will do some type of activity. Okay, so we talked about receptors and cell membranes in the previous lectures. All right, so the synaptic cleft is the space in between the pre-synapse and the post-synapse. And all of the neurotransmitters are stored in synaptic vesicles. And so if you remember also in previous lectures, this is a very specific type of transport. And it starts with an E, ends with cytosis. If anybody remembers what I'm talking about, what kind of transport is this? This is exocytosis. And so neurotransmitters, because they're such big molecules, or for the most part they are, not all of them. There's different types of neurotransmitters that are gases as well, but we're not really going to cover those. But for the most part, majority of them are transported by vesicles out, and they are exported out through exocytosis by synaptic vesicles. So neurons basically are doing their communication at the synapse. And the synapse, we already said, is a synaptic cleft. It's the one that's at the end of the axon. This is where all of the communication is going on, and this is also a major site for drug action. And so an action potential arrives at the synapse. The synaptic vesicles fuse with the plasma membrane. And so here we are with the synaptic vesicles carrying some type of neurotransmitter and releasing it out into the synaptic cleft. And so release of neurotransmitters go into the synaptic cleft. And neurotransmitters then bind to receptors on the postsynaptic cell. So this is the postsynaptic cell. These are the receptors. And once the neurotransmitter binds, it basically sends a signal to the next neuron, and it'll tell the neuron to fire rapidly, or maybe fire at the same pace, or slow down its firing. So it can change its rate, its frequency of electrical signal. And in some cases, the receptor channels open. They're called ion-gated channels, and that just allows the entry and the exit of different types of ions, which is like sodium and chloride and potassium and whatnot. And those ions basically help generate electrical signals. All right, now let's talk about the chemical messengers of the brain, which are called neurotransmitters, and the specific receptors that they bind to. Neurons communicate at synapses with neurotransmitters, and so we've already been covering that. We've been mentioning that over and over again, that neurons send a signal down their axon to the axon terminal and release the neurotransmitter to the next post synapse. And so that basically sends information down the dendrites to the cell body where the next signal can then be propagated. And in some cases, the axon terminals of a neuron can also be located, or they can be bound to blood vessels to also cause different types of changes, or they can cause the blood vessel receptors to open up their channels to allow certain ions to go in and out. All right, so neurotransmitters, chemical messengers of the brain that are basically located all over the central nervous system and the peripheral nervous system to generate action potentials, electrical signals. Neurotransmitters are communication molecules, and they can cause the brain to behave in different ways, either by excitatory or inhibitory. So if it's excitatory, it means that the neurons are firing at a fast rate. And if it's inhibitory, then neurons are slowing down. And some examples of excitatory are like when you're scared and you want to run from something, and so your neurons are firing quickly to generate these signals so that you can do that action. Or when you feel, when you take maybe like a sleeping pill and you start feeling sleepy and your central nervous system starts to slow down, that would be considered inhibitory because your neurons aren't firing as much. It's causing you to relax. We already said that I know I'm being totally repetitive here, but you'd be surprised. Repetition really helps with memorization. So hopefully, at least it's helping you with that. And so, neurotransmitters, we said, are released by exocytosis. The vesicles that carry the neurotransmitters fuse onto the cell membrane and release the chemical. So, in this case, you're looking at dopamine molecules that are basically binding to dopamine receptors. Now, every type of neurotransmitter has a very specific receptor. Not just any receptor can recognize that neurotransmitter. It's kind of like a lock and a key. You know, you can only use a certain key to unlock the door, and so it's the same thing. There's only certain neurotransmitters that can activate receptors. So receptors are the ones that are at the postsynaptic terminal. So this is the postsynaptic terminal. And this is an example of dopamine receptors receiving signals from dopamine. And so once they receive the signal, for example, this is a dopamine molecule binding to a receptor, then that sends a signal to the next neuron, and it'll tell that neuron to fire quickly. Now, these neurons, for example, they're located in certain areas of the brain, and I'm talking about the neurons of dopamine, are located in certain areas of the brain that are connected to the pleasure centers. And so the firing of this neuron means that it is activating the pleasure center. And there's also ways to reduce the dopamine in the synaptic cleft. And so an example here that you're looking at is an enzyme basically breaking down the dopamine so that it doesn't have that stimulatory effect anymore. Let's talk about how neurotransmitter is blocked, because having too much neurotransmitter at the synaptic cleft will just cause too much signaling for the next neuron, and it'll never really shut down. And so there has to be a way to regulate the amount of neurotransmitter that is available at the synaptic cleft. And there are several ways, and there are three that we're going to talk about. And so one is by the breakdown of with enzymes. And so there are enzymes that recognize a neurotransmitter, and they can basically just chop it up into little pieces and that are no longer functional, or that can no longer bind to the receptor and cause a signal. There's another interesting one called reuptake, in which receptors, let me draw in the receptor here, in which receptors are actually located at the presynapse. And so this is me pretending to draw a receptor. And what happens is the dopamine molecules are retaken, they're uptaken back into the presynapse, and they can even be recycled at this point. They can be stored and recycled. And so that's a way to reduce the amount of dopamine that's at the synaptic cleft. And the other one is by elimination, which is basically blocking. So we block here at this point. So the release of the neurotransmitter is blocked, and so it does not end up being released. And so those are ways to regulate the amount of neurotransmitter that is available at the synaptic cleft. Now, this is also a great way to create drugs, right? So this is a great way to create pharmaceuticals to regulate the amount of neurotransmitter available at the synapse. And so, like I said, this synaptic cleft is a major site for drug action, legal and illegal. And it happens to be that drugs, like for example, like THC, if you've ever heard of THC, that's a chemical that is found in marijuana. It happens to mimic a natural chemical receptor, or actually a natural chemical neurotransmitter that we have in our brain called anandamide. And so THC is basically like a mimic of anandamide, and it'll act on the same receptors as our natural receptors. And so basically what I'm trying to say is that drugs tend to target the synaptic cleft and tend to do things that are similar to what our neurons naturally do to regulate our own neurotransmitters. All right, let's take a closer look at the reuptake receptor, which I think is really neat. This is actually a great site for drug action. So let's talk about a legal one, which is called an SSRI. And an SSRI, which is a serotonin reuptake inhibitor, is used basically to treat depression. And so that's one type of drug that's used for that. But there are many types of reuptake inhibitors. So an SSRI basically blocks the reuptake receptor. And so if you block a reuptake receptor, what do you think will happen to the amount of receptor available at the synaptic cleft? Because earlier we said that the reuptake receptor takes the neurotransmitter back in to be recycled. But if you block that, what happens to the amount available at the synaptic cleft? Well, that amount available increases. So a serotonin reuptake inhibitor would block this transporter, and then you'd end up with a whole bunch of excess serotonin at the synaptic cleft. And if there's excess serotonin, that just means that the signal will continue propagating down the neurons, and so it'll basically continue to stimulate neurons. And there are drugs that have that similar effect too, like cocaine. From my understanding, cocaine also does this blockage. So it blocks a receptor that uptakes dopamine. And so you end up with a lot of dopamine at the synaptic cleft. And if you have excess dopamine, it's going to continue triggering the neurons that give you that feel-good effect. So here's an example of what I mean about about cocaine. And so here you have cocaine blocking the reuptake receptor in a dopamine neuron. And so here you are blocking the uptake of dopamine, and if dopamine is not being transported back into the presynapse to be recycled, then it's just hanging out here at the synaptic cleft. And if it hangs out here, it's basically stimulating these neurons over and over again, increasing the intensity of the effect, which gives that feel-good effect. Same thing with the SSRIs, like I mentioned earlier. These are the serotonin reuptake inhibitors. They basically block the reuptake receptors that are found at the presynapse, which means that there is more serotonin available at the synaptic cleft. And that also increases the intensity of the information that the neurons are receiving, causing them to fire over and over again. All right, so there are many different types of receptors. We're going to cover opioids, serotonin receptors, cannabinoid receptors, dopamine, cholinergic, adrenergic. I'm not covering every single thing on here, but this is basically types of receptors, just to let you know that receptors are very specific to their neurotransmitters. Not just any neurotransmitter can stimulate any receptor. It's like a lock and key. And we're also going to talk about the neurotransmitters that are basically the chemical messengers of the brain. We're going to talk about acetylcholine, epinephrine, which is basically adrenaline. I don't know if you've ever heard of the term adrenaline rush, where it makes you feel stimulated and like you have a lot of energy. And its cousin here, these two molecules basically are almost very similar, norepinephrine. And we're going to talk about serotonin, dopamine, GABA, glutamate, endorphins, and I forgot to add number 9, which is endocannabinoids. Now, the structures of neurotransmitters are different. Some of them look like amino acids, others look like whole proteins. Some are just gas molecules, like carbon dioxide can act like a neurotransmitter. So we're not going to cover all of the neurotransmitters, just some very specific ones. And these are the ones that basically drugs have the most effect on, or they either mimic these molecules. And if they're mimicking these molecules, that means that it, it can basically have the same effect on the brain as these type of neurotransmitters do. It's like an imposter. All right. So, like I said, adrenaline and noradrenaline are very similar in structure. And even if you look at dopamine, it almost looks exactly the same, except that it's missing its oxygen chain here. So it's a little bit different and has an extra molecule here. But it's interesting how some of these neurotransmitters are similar, but not all of them are. And so serotonin has these two carbon cyclic molecules. And here's acetylcholine, glutamate. Acetylcholine and glutamate are also very similar in structure. And there's endorphins and GABA. All right, and we mentioned earlier that neurotransmitters can have an excitatory effect or an inhibitory effect. If it's excitatory, that means that neurons are firing more rapidly. And if it's inhibitory, that means that neurons are slowing down. And it makes sense that our central nervous system and our peripheral nervous system has these two types of functions. And because if there's too much excitatory activity going on, there needs to be something that brings that activity down. And if there's too much inhibitory activity going on, there needs to be something that brings the activity back up. Okay, so it's sort of like a check and balance. All right, so let's talk about the neurotransmitter acetylcholine. The receptor for this is one type, by the way. There are different types of receptors, but a class of receptors that bind to acetylcholine is called cholinergic receptors. And the enzyme that breaks down acetylcholine, because remember we said that there needs to be something that regulates how much neurotransmitter is available at the synaptic cleft. And so one of the enzymes that regulates how much acetylcholine should be present at the synaptic cleft is called cholinesterase. And it basically degrades the neurotransmitter acetylcholine. And so acetylcholine is what we consider excitatory, meaning that excitatory and inhibitory, meaning that it has the ability, depending on where it's located, like in the brain and in the peripheral nervous system. So it just depends on what type of neuron we're talking about. But in some cases, it could cause the neurons to fire more rapidly, and in other cases, it can actually cause the neurons to fire more slowly. And that's because it also works with other neurotransmitters and it works with other ions to do this. They don't always just work alone. The neurotransmitter acetylcholine plays a role in the central nervous system, so we're talking about the brain and the spinal cord, and the peripheral nervous system, so we're talking about the rest of the body. And so in the central nervous system, it plays a role in memory formation, sensory reception, behavior, arousal, and attention span. So this is more of like an excitatory effect, whereas in the peripheral nervous system, it helps with the contraction of smooth muscles, slows down heart rate, and so this is an inhibitory effect, and dilates blood vessels. And so deficiencies in acetylcholine have been associated in patients with Alzheimer's disease. And so one of the major side effects of Alzheimer's is memory loss. And so people tend to have less acetylcholine, or people with Alzheimer's tend to have less acetylcholine in their central nervous system. And scopolamine, which is a psychedelic drug like LSD, tends to block acetylcholine receptors. And so one of the results is amnesia. And so people who take scopolamine tend to forget about their experiences because of this blocking of acetylcholine receptors. And acetylcholinesterase inhibitors, meaning that this allows this is inhibiting the enzyme that breaks down acetylcholase. And if you're inhibiting the enzyme that breaks down acetylcholase, that means that there's more acetylcholine around. And so there are drugs that are designed to inhibit that enzyme so that more acetylcholine can be at the synaptic cleft and hopes to improve memory. And so some of these treatments are for patients with Alzheimer's. All right, neurotransmitter norepinephrine. It is found in the central nervous system, and the receptors, and these are a group of receptors. And remember, there are many names, there are many different types of receptors, but this is just to give you an example, are adrenergic receptors. And an enzyme, one of the enzymes that breaks down norepinephrine, so that way we can regulate how much norepinephrine is in the synaptic cleft, is called monoamine oxidase receptor, which basically degrades the neurotransmitter. Neurons from the brainstem project widely to the cerebral cortex. So we have the brainstem here, projecting to the cerebral cortex and to the limbic system. Remember, we said that was the emotional centers, and the hypothalamus and the cerebellum. And so in these locations, you can find neurons that have the norepinephrine neurotransmitter and receptors. And norepinephrine is more of an excitatory type of neurotransmitter, meaning that it increases the frequency of the signal transmission. And a result is the increase in blood pressure and heart rate, and it produces that alerting feeling. Also, the positive feelings in the reward system and analgesia. Epinephrine is basically like the cousin of norepinephrine. It is mostly found in the peripheral nervous system, which is the rest of our body, not the brain and the spinal cord, but the rest of the neurons that are branching out to our body. It is basically adrenaline. So I don't know if you've ever heard of adrenaline rush, but it's that feeling of like excitement, or it gives you a lot of energy to do something. So like if you're, I don't know, like if you're in a fight, you need more energy so that you can defend yourself. That's an example. And so the receptors are adrenergic receptors, and these are the ones that basically recognize the neurotransmitter epinephrine. And the enzyme that breaks down epinephrine is also monoamine oxidase, and it basically regulates the amount of transmitter that is present at the synaptic cleft. And so this neurotransmitter is involved in the production of the fear stimulus, which causes that flight or fight response, meaning that if you're, let's say, for example, in a dangerous situation, are you going to fight it off? And so you would need energy to fight it off. Or are you going to run away from it? And regardless, you would still need energy in either case. And so epinephrine there's a link or a correlation in which it is produced in stressful situations. And it is also linked to the increase of blood flow to muscles, increased heart rate, and blood pressure. Okay, so as you can see, this right here is an excitatory type of neurotransmitter, which causes you to do some type of action. All right, let's talk about dopamine. And so dopamine is another type of neurotransmitter. The receptors are called dopamine receptors. Now, this is a general name, and so you can have D1 type of receptors, D2, D3, D4. So there are different types of receptors, but we're just talking about the main group of receptors. And an enzyme that breaks down dopamine is monoamine oxidase as well. And so it basically regulates how much neurotransmitter is available at the synaptic cleft. And so dopamine neurons are found in the brain stem. They project forward to the frontal brain, and also project down to the spinal cord. The effect of dopamine neurotransmitter is the production of feelings of pleasure, part of the reward circuit, and it also affects motor function, meaning muscular movement. Too much dopamine means there's too much pleasure reward system going on. So Overstimulating dopamine neurons gives that sensation of feeling good, of pleasure and reward, and that is also tied into addiction. And too little dopamine, it has been linked to depression, low motility, not wanting to do much, like lack of motivation, and it's also been associated in Parkinson's disease. In general, neurotransmitters affect the central nervous system differently depending on where these neurotransmitters and receptors are located. For example, if the neurons in the hypothalamus that have dopamine receptors are stimulated, those specific neurons are involved in the regulation of certain hormones. And schizophrenia patients that take antipsychotics, for example, those antipsychotics tend to block the dopamine in the hypothalamus. And they have noticed that patients that take these type of medicines to block dopamine tend to have fluctuations in their hormone functions. And so that's just it's like an indirect way of knowing that dopamine in the hypothalamus regulates certain hormones. If the dopamine neurons are located in the substantia nigra, which is here, it's like a little location here, then those neurons are basically involved in the regulation of movement. And so people with Parkinson's tend to have low dopamine levels in that very specific region in the substantia nigra. And antipsychotic drugs, which block dopamine, we already said they block dopamine, tend to have what we call a Parkinsonian effect, which means that they can be shaky, they can have a hard time moving because there's little dopamine going on in the substantia nigra. There are also dopamine neurons in the ventral tegmental area. And these neurons are located at the VTA and project out to the frontal cortex and limbic system. And remember, we said that the limbic system has to do with the emotional center of our brain, and so that helps us produce emotions. And so dopamine neurons affect that system as well. And it has been shown that alterations in the development of this pathway may be involved in schizophrenia, meaning that if the neurons here are not necessarily at their optimal functioning, so if there's something going on with the dopamine levels, specifically in this area, that has been tied into schizophrenia. And so in this case, it's actually research suggests that it's too much dopamine activity going on. And so that's why the drugs that are used to treat schizophrenia tend to block dopamine receptors, thereby reducing the amount of dopamine available and reducing the basically the stimulatory effect that dopamine has on that system. And this is also an area, so the VTA is also a major area in which drugs activate. And so when drugs activate that area, it tends to produce sensations of pleasure, which is also involved in addiction and drug abuse. And so notice, depending on where the neurons are located in the brain, that it can have very different effects on our overall behavior. There are a class of drugs called antidepressants or antipsychotic drugs, which are basically MAO inhibitors. And so that means that it's monoamine oxidase inhibitor. And remember, we said that that's the enzyme that breaks down dopamine. So if you are inhibiting that, meaning that if you are blocking that, that basically increases the amounts of dopamine available. And so some antidepressants focus on that to increase the amount of dopamine and norepinephrine just because they act on the same neurotransmitter that is available at the synaptic cleft. Let's move on to the next receptor, which is serotonin. And so serotonin was first investigated when there was studies of LSD going on. And serotonin was of interest because it is somewhat similar in structure. So if you notice the red part here of the serotonin to LSD, and so it matches this area here. And so, because of that, because LSD kind of looks like serotonin, it was believed that LSD tends to act on the serotonin neurons. And also, they hypothesized that perhaps the hallucinations in LSD had to do with the alterations of the functioning of the serotonin neurons. And so, meaning that LSD was sort of messing with the activity level of the neurons that are activated by serotonin. The receptors for serotonin is just this is just a general broad name. It's called serotonin receptors, and again, there are many different types of receptors that fall in this class. Um, and the enzyme is also monoamine oxidase, and so this also can break down serotonin to keep the levels of serotonin at the synaptic cleft in check, basically. And so serotonin is known to play a role in mood, depression, anxiety, obsessive compulsive disorder, panic, phobia, sleep, sex drive, cardiovascular function, regulation of body temperature. Normal levels of serotonin help regulate mood, social behavior, appetite, digestion, sleep, memory, sexual desire, and function. And low levels of serotonin have been linked to depression, poor memory, difficulty sleeping, anxiety, food cravings, obsessive compulsive disorder, panic, phobias, lack of sleep, and low sex drive. And so there are a class of drugs that are targeted to increase the amount of serotonin available at the synaptic cleft to treat these symptoms, especially like depression. Serotonin neurons, they are found throughout the brain, in the upper brain stem, in the cerebral cortex, so they project out here. They project out to the hypothalamus and throughout the limbic system. And so we know that the limbic system is associated with our moods and our feelings. Earlier in this lecture, we talked about an antidepressant called an SSRI, which is a serotonin reuptake inhibitor, meaning that it prevents serotonin from being reuptaken into the presynaptic cell. And so, by inhibiting, by blocking the amount of serotonin that gets sent back into the presynaptic neuron, you are allowing for more serotonin to hang out at the synaptic cleft. And if there's more serotonin available, then that serotonin can continue stimulating these neurons. And if they are stimulating these neurons, then that can help with our mood. So that can stimulate the parts of our brain that are associated with anxiety and sleep loss and depression. Okay, basically having more serotonin around can continue firing those neurons that need to be fired. Maybe those neurons are not stimulated enough. And so by having these serotonin reuptake inhibitors, you can increase the activity of those neurons. All right, so the two major pathways, especially these are the pathways that drugs have a very heavy effect, you could say, a profound effect, are the dopamine pathways and serotonin pathways. So just remember that. And also notice that some of the tracks from the dopamine pathway tend to be in the same areas as the serotonin pathways. But yet the serotonin tends to extend more throughout the brain, hitting the limbic system. And so dopamine pathways are the ones that are associated with reward, pleasure, motor function. Remember, that's muscular function, compulsion, preservation. And serotonin pathways are associated with our mood, memory, sleep, processing, cognition. And so, again, these are the major areas in which drugs target, which is why people tend to have mood swings, especially people with addiction, because these pathways are being tampered with. Let's talk about GABA, which is another neurotransmitter, short for gamma aminobutyric acid. Notice in the name that it's called amino because GABA is an amino acid, and interestingly, it is also a chemical messenger of the brain. The receptors that receive signals from GABA are simply called GABA receptors. And so these receptors are capable of receiving the signal or capable of binding with the neurotransmitter GABA, which then sends information to the next neuron. And GABA is considered a major inhibitory neurotransmitter found throughout the brain, so meaning that a lot of neurons use this neurotransmitter to slow down activity. And so inhibit means to prevent, so in this case, it's slowing down the activity, and so it reduces neuronal excitability. Commercial drugs that mimic GABA or stimulate the release of GABA are used to treat anxiety and epilepsy. So, if you think about it, in epilepsy, the neurons are firing too rapidly. And so, if you have more GABA around, that can help the neurons slow down and to not fire so quickly. And same thing with anxiety. So, anxiety can give this increase of energy feeling. It can give this anxiety, this antsy feeling. And so the same thing, a person needs to feel relaxed. And so anti-anxiety medications like benzodiazepines act on GABA, act on the GABA system to increase the amount of GABA available so that the neurons can relax and quit firing. And so it also serves as an anesthetic, and it's also found in other drugs, for example, like drugs associated with bipolar disorders to basically relax the neuronal firing of certain regions of the brain. Glutamate is another type of neurotransmitter. It's basically like the cousin. It's very similar to GABA. Well, very similar in structure, but not in function. So, glutamate is basically glutamic acid. The receptors are a class of receptors, and some examples are NMDA receptors and AMPA receptors. And there's not necessarily enzymes that break glutamate at the synaptic cleft. What there is are transporters that basically transport glutamate. And glutamate and GABA, they are so similar that there are enzymes that convert glutamate to GABA and GABA to glutamate. And so they're kind of just recycled. And so that's why they're transported around. And so glutamate is the exact opposite of GABA, in which it's the major excitatory neurotransmitter, meaning that it increases the activity, it increases the amount of firing. And so this is located in like 50% of your brain. So it's one of the major excitatory neurotransmitters, where GABA is the major inhibitory neurotransmitter, doing the opposite effect. One is relaxing while the other one is increasing the activity. And so GABA has been linked to strengthening synaptic connections and also in learning and memory. High concentrations in these, there are high concentrations of glutamate neurons in the cerebral cortex, in the hippocampus, basal ganglia, septum, and amygdala. So these are some of the neuronal tracts that carry GABA neurotransmitter and GABA receptors. And so like I said, this is basically a neurotransmitter that causes neurons to fire even more frequently. There are certain drugs, for example, that block glutamate receptors like PCP and ketamine. And by blocking it, it gives a more like relaxing effect, right? Because these are excitatory neurotransmitters. So if you block these neurotransmitters, then you get less neuronal firing, which is a relaxing state. Too much glutamate, in fact, can be very toxic to neurons. It can actually cause them to die off. And so you'll see an interesting side effect in alcohol withdrawal. So alcohol actually reduces the amount of glutamate available. And in alcohol withdrawal, so if a person isn't drinking alcohol, the neurons tend to try to compensate for the amount of glutamate that wasn't there. And so it'll just produce a whole bunch of extra glutamate, and this will in turn end up damaging neurons. Just because it's trying to compensate for the loss that it had when the person was drinking alcohol. Increased levels of glutamate, as you can imagine, relate to epilepsy. And so remember, epilepsy is the neurons that are firing quickly. They're kind of out of control. And so an increase in glutamate has been linked to it, has been linked to epilepsy and hypoglycemia. And some drugs like PCP that block glutamate have been known to cause hallucinogenic effects and euphoric feelings. Let's talk about our next set of neurotransmitters, which are opioid neurotransmitters, and they're also called opioid peptides because they look like whole proteins, not like GABA and glutamate, which look like little tiny amino acids. Opioid peptides tend to look like bigger molecules. And some examples are endorphins. So if you've ever heard of endorphin rush, or if you've ever, let's say for example, you hit your hand, it hurts at first, but then after a while it feels kind of good. That's that endorphin rush. It's the endorphins that are preventing the feeling of pain. And so some opioid neurotransmitters are called endorphins and enkephalins. And the receptors for these neurotransmitters, and remember we said that the receptors are the ones that receive the signal from the neurotransmitters, are called opioid receptors. And the enzyme that basically breaks down the neurotransmitters are called peptidases. These type of neurotransmitters have been associated with pain perception, the feeling of reward, and so reward is that feel-good feeling that causes the behavior of repeating that feeling so that you can feel it over and over again. It's also associated with emotional stability and energy highs. Opiates such as morphine, codeine, and heroin tend to activate the opioid receptors, and so if these opioid receptors are activated, then that means that the feelings of reward and emotional stability and energy highs are increased. And so that's why morphine, codeine, and heroin are one of those really popular and addictive drugs, simply because they're acting on the opioid receptors, which release more of that feel-good feeling by stimulating the neurons that are associated with that. Let's talk about the last type of neurotransmitter, which is called endocannabinoids. And endocannabinoids are a class of neurotransmitters. An example of one is called anandamide. And these are neurotransmitters that your own brain produces. And there's a very popular molecule out there called THC, which is found in marijuana, that tends to look very similar to anandamide and causes the same effects as our own natural neurotransmitters. And so endocannabinoids bind to the receptors called CBRs. So these are cannabinoid receptors, and some examples are CB1 and CBD. And so there are many different types of receptors, but these are just examples. And this is an interesting kind of neurotransmitter. For one, it is lipid-based, and so meaning that it resembles more like an oil or a fatty. And remember, we said that the cell membrane is readily receptible for other molecules that are like it. And so when we say lipid, we're also referring to nonpolar. And so this is a type of nonpolar molecule that can easily interact with the cell membrane and easily make its way into the cell. And endocannabinoids are found throughout the central nervous system and the peripheral nervous system. For example, in this diagram here, the CB1 receptors, which bind to endocannabinoids, you can find them throughout all of the peripheral nervous system and in the brain. And CB2s tend to be more found in the peripheral nervous system, and so you don't see many of those receptors in the brain. THC is a molecule called tetrahydrocannabinol, and that is found in marijuana, and it's very similar in structure to an endocannabinoid. And so that means that because it's similar in structure, that means that it can activate the same receptors as the endocannabinoids do. So it can activate the CB1s, the CB2s, the CBRs, and cause the same type of effect. And so endocannabinoids have been associated with appetite, pain sensation, mood, and memory. And for example, THC is associated with an increase in appetite and memory loss because of the stimulation of those CBR receptors. And if you look at the left here, it's interestingly, THC is not too similar in structure as anandamide, but yet can act on the same receptors. And so they have some structural similarity, which allows them to interact with the same type of receptors in the brain. These are your feel good neurotransmitters like dopamine, serotonin, endorphins, and oxytocin. Oxytocin is that molecule that is associated with feelings of love and closeness. And there's an increase of oxytocin when a female gives birth. And here's just a little picture for you to look at so that you can look at all of the neurotransmitters in one slide, and you can just study how each one has a different effect on the brain and even in the locations that are mostly found in.